Okay, so today we're going to talk about cells and the structure of cells. We're going to look at prokaryotes and eukaryotes, but we're going to start all the way back with the basics first. So, cells were kind of first discovered by Robert Hooke, who was an English scientist, and later, after discovering that, many scientists discovered that everything was made of cells, they came up with what was called cell theory, which we may have mentioned before in some of the previous lectures, the introductory lecture, I think. And the two scientists were Schleiden and Schwann, and I'm not sure if I'm saying those right, but they concurrently came up with this theory called cell theory. And there's three parts to it. And the first part is that all organisms are made of cells. And within those cells you have metabolism occurring, where things are breaking down stuff for energy, and they are able to inherit things from other cells. Okay, so that also occurs in cells, and that we know, of course, that's through the process of genetic transfer of DNA, or the duplication of DNA. The second part is that cells are the smallest living things and the basic units of all living organisms, and so this goes on to kind of define that living things are made of cells if all living things are made of cells. It's kind of like we stated that. So this kind of excludes then viruses as being alive because they aren't made of cells. The third part is that cells come from the division of other cells. Okay, they don't spontaneously arise from a bunch of macromolecules, but that they must split into two different cells. So some things about cell size. It's, one question you might have in thinking about cells, well, why not just be one big cell? Why do we have so many little cells combining to make larger organisms? And it has to do with the rate of diffusion by which things go in and out of a cell. So we are limited in our cell size because of the rates of diffusion. And rate of diffusion is affected by surface area available, temperature, concentration gradient, and distance. So if you have two cells, one of them is big and one of them is small, and our little red thing there is our molecule trying to get through the cell, it can diffuse through both of these cells, but cell 1 has a much further way to go to get to the center of the cell, whereas cell 2, it's a closer distance. So things are going to diffuse better through a bunch of smaller cells than they are through a larger cell. Okay, and this is a figure from your book which kind of reiterates that. If you have two cells that are, one magnitude order of difference, so 1 unit and 10 units, the difference in surface area is quite big, right? The surface area to volume ratio is about 3 in the thing with 1 unit and is about 0.3 in the thing with 10 units. So there's much more surface area, which means much more area for diffusion to take place in a smaller cell than in a larger cell, and that defines how big cells can be. So in order to see cells or to see parts of cells, we need microscopes. Okay, and that increases then the level of organization by which we can study things. So before microscopes, we could only study things at this rudimentary organismal level. You could kind of go into what different tissues were, you could do organs, but cells were too small to see. So what microscopes do is they increase the resolution, or increase our ability to differentiate between two different objects. So down here you see, here are two very, very small objects. These are what make up flagella, and you can see them differently. Whereas, obviously to the naked eye you wouldn't be able to. The compound light microscopes are the ones you're used to using in lab, and they use two lenses and a light source from below to illuminate the specimen that you're looking at. And the resolution on them is 200 nanometers. An electron microscope has a much greater resolution, 2 nanometers, and it uses a beam of electrons and film behind it to make an image, and that's what's done here in this one. The scanning electron microscope does a similar thing, but it is better for surfaces. So this is done by a scanning electron microscope of an ant. The transmission electron microscope beams through materials. So this over here is a transmission electron microscope. So when looking at cells, there are some common themes. So all cells have DNA. All cells have a nucleoid or nucleus, a place where the nucleus has concentrated. And then all of them have ribosomes, which are important for important for protein synthesis. All of them have a cytoplasm, some sort of fluid matrix on the interior of the cell by which things kind of float around. It can be transported throughout the cell. And all of them have a plasma membrane, which is a lipid bilayer, okay, embedded with a bunch of proteins and other molecules which help the cell function. Prokaryotes are one of the types of cells that we have, and are also a taxonomic differentiation between different types of cells. So organisms that are prokaryotes are single-celled, so they do not have double. They cannot make organisms of multiple cells. They do not have a nucleus, so instead they have a concentration of DNA at the nucleolus, and they have a cell wall made of what's called peptidoglycan, okay, which is part sugar and part proteins. Peptide stands for protein, glycan stands for sugar. The cell wall is important for protecting them, also can help them go in the dormancy period. And it's different than a cell wall of plants and fungi because it's made of a different substance. They do not have organelles other than ribosomes, which kind of freely float within the cytoplasm. And they do not, some of them do have accessory structures such as cilia, like you can see on this one, although this is a eukaryote. Or they may have a flagella coming off of them as well. There are two domains that are prokaryotes, and they are archaea and bacteria. Eukaryotes, then, on the other hand, have a membrane-bound nucleus. Okay, so the DNA is found within one area. They have membrane-bound organelles and a cytoskeleton, so an internal network of proteins which helps hold their cell together. They also have what's called an endomembrane system, where they have these folded membranes. So you can see here and here and here, all part of the endomembrane system, where they can move things throughout the cell, put membranes around things, package things, and help specialize different parts of the cell into different functions. Alright, so now we're going to go through the different organelles, starting with the nucleus, which is the most, I shouldn't say most important, but it's kind of like the part that has all the genetic information, all the blueprints of how to do everything. Okay, it holds DNA in linear structures called chromosomes, and it has a concentration of DNA at the nucleolus, where ribosomes are synthesized. So you have a lot of enzymes and enzyme activity happening there. At the nuclear envelope, which is what surrounds the nucleus, has two lipid bilayers and pores, you can see those here, by which substances can go in and out of the nucleus. Alright, ribosomes, these are not technically organelles because they are just made of RNA, ribosomal RNA, and they are important for synthesizing proteins. There are two types of ribosomes. You can have free ribosomes, which are floating in the cytoplasm, or you can have rough endoplasmic reticulum which is where ribosomes are stuck to a structure, which we'll talk about later, the endoplasmic reticulum. Okay, there are two types of endoplasmic reticulum: rough ER, which has the ribosomes on them, and smooth ER, which has a variety of functions, including storage of calcium or lipid and carbohydrate synthesis. It can also help to detoxify substances. Okay, so you can tell the difference though, because rough ER has these dots on them, and smooth ER does not. The Golgi apparatus then is another part of the endomembrane system. It's composed of flattened stacks of interconnected membranes, and they function to transport molecules within the cell or outside the cell in vesicles. So you may have, and also you have, the cis side, which is where molecules go in, or where maybe a vesicle goes in, then it releases some substance which then is modified within it, and then vesicles are formed on the opposite side, on the trans side, where you can have then different vesicles formed. Okay. All right, lysosomes then are formed by the Golgi apparatus, and they are a membrane-bound concentration of enzymes which are used to digest old or damaged organelles, and they can also be used to digest food, or also maybe if you have some sort of virus or bacteria, it can also help to digest and break those down. Perioxisomes are another vesicle which is formed that contains an enzyme within them, and the enzymes are important for breaking down hydrogen peroxide into water. Hydrogen peroxide is a very volatile substance which will hurt other tissues or hurt other parts of the cells, damage other parts of the cells. That can also contain enzymes that help oxidate fatty acids, break them down so you can use them for energy. Vacuoles, these are only found in plants, and they're big membrane-bound structures. You can see this vacuole here takes up a lot of portion of this cell. They're important for helping keep the shape of the cell so they can take on water or release water if they want to make the cell tight or limp. And then they can also store different types of things like water or carbohydrates or food molecules or other things as needed. Some fungi and protists also have contractile vacuoles, which can help change the shape of the cell. Okay, mitochondria, these are basically the powerhouse of the cell. So this is what's going to provide energy for all the other parts of the cell. And the process by which energy is made is called oxidative phosphorylation, which requires oxygen. It has a double membrane, okay, so one here and a folded inner membrane. And then they have their own DNA. So separate from the DNA which is in the nucleus, and when they divide, they pass their DNA onto the new cells. And this mitochondria is passed on from father to child, or from parent to child through the mother. So all the mitochondria you have came from cells that divided originally from your mother. Alright, chloroplasts then are where photosynthesis takes place, and this is found in plants and cyanobacteria. They contain pigments called chlorophyll, or one pigment called chlorophyll, and other pigments as well. And it's excited by sunlight. That when it is excited, it can trap that energy to make sugars. It also has two membranes similar to the mitochondria, and they have their own DNA as well. The inner membrane is composed of stacks called thylakoids, and those are your chloroplasts. So one of the theories with eukaryote cells is that not all the organelles were originally from one cell. In fact, the theory of endosymbiosis is that chloroplasts and mitochondria actually originated as separate organisms, which somehow got incorporated into a larger organism, and just by chance they were able to survive together and then eventually became dependent on each other. And the evidence for this is both have double membranes. Okay, why would they have two membranes if one would suffice? If it arose within a cell, you would expect it just to have one membrane. But the idea was that when they were first enveloped, they have their own membrane, and then as it went in, it captured part of the other membrane, and that's why it has two. They also have their own DNA, their own ribosomes, and then somehow evolved this symbiotic mutualistic relationship where both benefit. The cell can provide oxygen for the mitochondria and take care of its waste. The mitochondria provides energy for the rest of the cell. Similar with chloroplasts, which provide sugar molecules for the cells. All right, the cytoskeleton, like I said before, is a network of proteins. They have differing levels of thickness, but all of them help to shape the cell and give it structure, and may also be used to transport things back and forth throughout the cell. So microfilaments are the smallest, microtubules are the largest. So we have a microfilament called actin, a microtubule here, and then intermediate filaments are intermediate in size. Okay. The centrosome is a region surrounding centrioles, okay, and this is contains microtubules that guide chromosomes during cell division and also make up flagella and cilia. So these are important for when we talk about cell division, and it'll make more sense when we talk about it. Plants and fungi lack centrioles, so this is specific to animals. Cell walls are found in some cells as well. Plants, fungi, and some protists have the cell wall. Plants and protists have a cell wall made of cellulose, where fungi has a cell wall made of a different substance called chitin. Now, outside the cell, there is also lots of projections and things which are still attached to the cell, but kind of come out, as you can see here, kind of like hairs. They're often made of glycoproteins, glyco referring to sugar and protein referring to protein. Proteins are embedded and then these sugar strands coming out of it. Or they could also be collagen, which is another protein in multicellular organisms. And this can form a protective layer outside the cell, can be really sticky and keep other organisms or bacteria or other things from coming in and infecting the cell. So here's a table from your book which shows prokaryotes, animal and plant cells side by side. These of course are both eukaryotes, and I just circled the things which were unique to one or the other. Okay, so finally we're going to talk about some cell-to-cell interactions which are important for first recognition of cells. So you want to make sure that a cell is going to be friendly and not trying to eat or kill you. So this is done by recognizing surface proteins, okay, proteins with a specific signature that identify you as safe. Glycolipids, these can also, of course, this refers to a sugar lipid, which can embed into the lipid bilayer and then have a sugar tail, and these can be unique to different cell types. The MHC proteins are also used by the immune system to recognize foreign cells. And we'll talk about what that is in class. So finally, you have connections between cells within the same organism, so in a multicellular organism, that are important for different things. So one of them is tight junctions, and this is important for making a waterproof barrier so you don't have water leaking through different cells. Okay, so your skin has a lot of tight junctions on it. Some areas need very strong connections. So an example of that are in your heart, and they use what's called desmosomes. This forms like a rivet which connects cytoskeletons of these adjacent cells. So you usually find these where you have a lot of force, a lot of repetitive force occurring. So heart cells are constantly beating, so you constantly have movement, and requires a little extra security in these desmosomes than the tight junctions can offer. The third one is gap junctions or plasmodesmata. So both of the plasmodesmata are in plants, and gap junctions are in animals. And these connect adjacent cells in their cytoplasm to allow ions to transfer between them, or hormones to allow for cell-to-cell communication. Alright, so here's an example of a plasmodesmata between two cells that is allowed to go through the cell wall and can then function to transport different chemicals that can be used as communication.